like to introduce our speaker?
Well, thank you very much. It is a pleasure to introduce Arthur, no stranger to Rotary. He is a director with the Rotary Club and has been a director for some time. He is the chair of the Community Service Committee, and he has a background in science, and we look forward to hearing what he has to say today in terms of illuminating the challenges that is not just a part of Canada, but is the global pandemic COVID-19. And as much as we at Rotary share a laugh and share a joke, I'm pretty sure he's going to give us some solid information today that's going to give us some sober reflection. So with no further words of introduction, Arthur, you have the stage.
I gave this title COVID-19 because we really don't know where the hell it's going to take us to. The future is just totally uncharted, and every day is a new discovery. And every day there are new information that comes from various sources from around the world. Let me first start about viruses in general. Now, viruses are a very simple form, the simplest form of life, and they can live only within living cells. They have a basic genome, which consists of either a strand of DNA or a strand of RNA wrapped in a protein vesicle. They use the host cell's machinery to reproduce and propagate. Viruses are supposed to have originated in vesicles near the hydrothermal vent in the primordial ocean floor, alongside the hypothetical last universal common ancestor. Now, this is a hypothesis because it's like which came first, the chicken or the egg? We know the virus cannot exist by itself. You need it to have a cell. So the best explanation that scientists, virologists think is they co-evolve, they evolve together. And as you see these bubbles coming around the hydrothermal vent, these are vesicles. And the vesicles are formed in various ways. They figure the virus formed in one way, and the virus got into the primordial cell, the LUCA, or the last universal common ancestor. And that's how they infected the first cell. Now, let me move this one. Now, if all the viruses, so 10 to the power of 31 on Earth were laid end to end, they would stretch for 100 million light years, which is the number of viruses we have. Viruses infect every form of life, from bacteria to fungi to various types of plants and all animals. And there are a whole host of them, various types of viruses. And of course, today we're going to talk about one such virus. Now, before I go into it, there's some basic terminology. You don't have to remember everything, but just have a rough idea. I'll tell you which one you should remember. You know, we all heard about the DNA double helix, Watson and Crick's model of the two strands of DNA. And they consist of what they call bases. There are various kinds of bases. And you have one sort of strand extends from the 5' to the 3'. The other one goes in the opposite direction from the 5' to 3'. And they are both connected or sort of held together by bonds, hydrogen bonds, and they form this helix. Now, the DNA can replicate itself because it's a kind of DNA polymerase, and then it is transcribed into RNA. This is the first step, transcription of DNA to RNA. And this RNA also can replicate by itself in certain forms, but it translates the message that is received from the DNA into proteins. It has got 21 amino acids to select from. And all these bases 3 of them code for an amino acid. So this is called the triplet code. For example, the amino acid glycine is coded by GGU. So we can do all these things even artificially in a artificial system. So DNA, transcription to RNA, translation into protein. Now during this translation, you see these 2 strands. One is called the sense strand. The other is called the antisense strand, which goes in the opposite direction. The transcription is by the antisense strand of the DNA into a sense strand of the RNA. Now, we don't have to worry about remembering all these things, but one thing that you remember is there are some viruses called retroviruses like HIV. They have an enzyme called reverse transcriptase because they have an RNA virus. It's got an RNA strand. What it does is, upon infection of a cell, it reverse transcribes the RNA into DNA. And it's from the DNA, it starts propagating and doing all the damage that it causes. So that's a reverse transcriptase. This was discovered about 10 years ago, the reverse transcriptase, and it's become a very useful tool in doing molecular biology. So this is just what you have to remember. COVID-19, the virus itself, COVID-19 is a disease. The SARS-CoV-2 is the name of the virus. Now, that virus is an RNA virus, and there's a single sense strand. And the RNA is highly unstable and can be easily destroyed in extraction. RNA viruses are generally unstable and mutate into different strains. Fortunately, COVID-19 is relatively stable, and therefore we can expect to make a specific vaccine. The flu virus, for example, mutates so much that every year a new vaccine has to be made. Now, this is a diagram of the COVID virus. It's called a coronavirus because it looks like a crown or a corona around the sun. Coronavirus is a large group of viruses that specifically infect the respiratory tract and range from mild cold virus to deadly SARS, MERS, and COVID-19. We have heard all of those. SARS is quite familiar to Torontonians, and MERS is a Middle East virus, and now COVID-19 has hit us again. They all have a corona or a crown-like appearance. The official name of the virus is Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV-2. And the disease itself is coronavirus disease, or COVID-19. Now, the virus itself is sort of a circular virus. It has got a lipid membrane. Now, that is where the washing with the soap comes in, because soap destroys this lipid membrane. That is why you use soap to wash your hands. Enclosed inside, you find an envelope protein, and then a genome which consists of RNA. This is a sense RNA enclosed in a protein. And then you have spikes on it. These are called spike protein. This is very interesting. This is what we should remember, because this is what the virus uses to get into cells. Infection is by inhalation of micro droplets containing the virus. The spike protein of the virus attaches itself to a receptor on the surface of the respiratory cell, or any cell lining the respiratory system. Now, this particular receptor is called angiotensin converting enzyme receptor, or ACE2. Now, this ACE2 is on the surface of all the cells lining the nose, the windpipe, the bronchial tubes, and all the way to the air sacs at the end where oxygenation takes place. Now, as soon as it attaches itself to this ACE2 with that spike protein, it is endocytosed, or it is sort of engulfed by the surface membrane, and it enters the cell. That's where the entry is made. And upon entry, the release the single-stranded RNA, sense strand. It is supposed to be 30 kilobases in length, which is fairly small. And as soon as it is released, this enters the host cell, the human cell's ribosome, where it replicates. For the replication and everything, it uses the host energy. The ATP, the energy source from the cell, supplies the energy for all the activities. And then there are a few proteins that are translated into viral proteins. So it goes into the cell. Now, it goes all the way into the lungs, right into the alveoli. And there it goes and attaches itself to the bronchial epithelial cells. So you have a lining, and that's the mode of entry. Now, the origin of COVID-19 is rather disputed. Some people think it's a sort of an accidental release of the virus from a virus lab in Wuhan. Wuhan has two virus labs. One is a high security lab that was established in collaboration with the French. And this lab, they've been using this lab to study highly pathogenic viruses and bacteria. The Ebola, for example, they studied there, and that's a high security lab. Some people think it came out of this lab and it's accidentally released. Now, this is one of those conspiracy theories. More likely is that the virus came from one of those wet markets where they sell juicy bats for people to eat. And I found out that bats are consumed not only by the Chinese, but from the Philippines, the Indonesians, lots of people in Asia consume these bats. I have never thought of the bat as a delicacy because it looks so ugly, for instance. But on the other hand, we eat lobsters, don't we? They're not actually very pretty. Now, the sequence of the RNA strand they have studied, and there's a similarity between the bat coronavirus and COVID-19. They're very similar. So they say it's probably the bat coronavirus came into the humans. It's what they call a zoonotic virus, jumping from an animal into a human. The ACE2 receptor, which needs to enter the cell, is present both in bats and in humans. So it's a very good argument that it came from the bats. Now we know the bat has a lot of these coronaviruses. Now, in the Middle East respiratory virus, it came through the camel. A close contact with mucus or fluids of the camel went into the human host. The SARS supposedly came through a civet cat. A civet cat used as a diet or a close contact, and it went to humans. The SARS-CoV-2, apparently, they say, came through the pangolin. It's sort of a scaly anteater. It's a sort of a threatened species, and it has to be protected, otherwise it will go extinct. And then it came to the human. Not exactly, they don't know exactly how it did. But one of the side benefits is, ever since they described this, people have left the pangolin alone. So it is not going to be a threatened species. So it's happily propagating and living a good life. Now, the coronavirus infection, it can be, sort of, they have a flu and allergies. They all have similar symptoms. Fever, cough, shortness of breath, symptoms appear 2 to 14 days after exposure. Also loss of smell and taste. These are all the coronavirus things. Flu, on the other hand, is fever, cough, muscle aches, fatigue, chills and sweat, congestion, and sore throat. So that's a bit of a difference that you have to find out. Allergies, again, during this time of the year with all the pollen, sneezing, itchy nose and eyes, roof of the mouth, runny nose, and all that stuff is happening. But you can unmistakably identify coronavirus because of the shortness of breath and difficulty breathing. But that happens after a while. Now, respiratory droplets fly in, not the fine particles that float away. It has to be a fairly big droplet that is, you breathe in. You can touch objects where the virus is, and then you can touch your face and get it too. So they have close contact within 6 feet. That's what people have established. Now, the virus itself has different kinds of severity. 80% of the infection is fairly mild and people recover. 13.8% severe. 6.1% is critical. Now, 6.1% critical is the one that older people are concerned, and also people who have underlying conditions like heart problems, diabetes, various types of kidney problems and things like that. Now, this is the pathophysiology of COVID-19. Nose and throat rich in ACE2 receptors hijacks the lining cell's machinery and makes myriads of copies right in the nose. It's asymptomatic for up to 14 days, but the person feels infectious. So you may not know that you have the COVID virus, and still you will be transmitting the disease. Fever, cough, sore throat, loss of smell, taste, head and body aches, these are all symptoms. Now, one of the things that people can readily check is their temperature. So if it is above 37.5, you have to worry. So I think I quickly went and bought myself a thermometer, and I checked my temperature morning, afternoon, and evening to see that I'm within that range. It moves down the windpipe or the trachea to the lungs, infects air sacs, which are extremely rich in ACE2. Immune cells invade, release chemical messengers called chemokines, kill virus infected with cells, fluid and dead cells accumulate, disrupt oxygen transfer, pneumonia, and rapid shallow breaths. Some recover with oxygen through the nose. Others deteriorate to acute ARDS, or acute respiratory distress syndrome. X-rays and computerized tomography reveals lungs with white opacities, alveoli filled with fluid, dead cells, mucus, and detritus. So what is really happening is the fight of our own white blood cells with the virus and destroying all the infected cells that causes the trouble in the alveolus. But it also affects all sorts of other things. It doesn't confine itself, because there are a few of these ACE2 receptors in other places. In the eye, it can cause conjunctivitis. In the brain, it can cause inflammation and confusion. And then in the liver, depress the enzyme. Kidney, it can cause failure by plummeting the blood pressure. In the gastrointestinal tract, it can cause diarrhea. Globally confirmed cases as of this morning is 2,582,529 infections and 178,481 deaths. That's from CNN information this morning. Now, the COVID destruction of the air sacs is perhaps the most important problem with the pathophysiology. Now, this is on the left, you see the air sac. And if you look at a healthy air sac, the air sac is surrounded by capillaries. And this is where all the branches of the lungs and the bronchial tubes branch and rebranch and rebranch, and finally end up in these air sacs where the oxygenation takes place. This is the alveolus, and it's got two types of cells. One is the lining cell, which is a type 1 cell. Another cell, type 2 cell, is the alveolar cell. And there's oxygenation and removal of carbon dioxide that occurs here, which is a normal one. But when the infected one, you have destruction. The sequence of events in the air sacs, this is what happens. Infection of alveolar cell, inflammatory signals recruit macrophages. These are all white blood cells that release chemical messengers, cytokines, that cause vasodilation. Now, and then there is, at this time, you get fever and cough. In other words, it's fairly mild at this time. More immune cells arrive and for destroying the virus. Fluid accumulates, which dilutes the surfactant, leading to the collapse of the alveolus, decreasing gas exchange. That time you suffer shortness of breath. Then you have another type of immune cells that come in, the neutrophils recruited, that release reactive oxygen. that destroys the infected cell. Now, at this stage, when it destroys all the alveoli, you need a ventilator. You suffer from acute respiratory distress syndrome. Now, there are two ways of testing the presence of the virus. The one is the antibody assay. Now, the best thing is the antibody assay is to use one of the proteins from the virus, usually the spike glycoprotein. That's the yellow one right on the little sort of outgrowths outside this. That's a spike glycoprotein. Now, the actual way an antibody is produced is you take this antigen, which is a protein, and you inject it into a rabbit. And the rabbit fights against this antigen and produces antibodies. You collect the antibodies, so you have an antibody specific to this antigen that you introduce. In the case of the virus, use the spike, and then you inject it into a rabbit, and then it produces primary antibody. And then when you want to test it, what you do is you take this primary antibody that you have created against the spike glycoprotein, attach it to a second antibody with a conjugated enzyme. Now, this enzyme is usually horseradish peroxidase, any kind of enzyme. The reason you do this, you magnify the detection system. The moment you have an enzyme, you put a little substrate, the enzyme converts it into an end product, and the end product is usually colored, and you can measure it. And so you have a lot of these substrates becoming end products because of this conjugated enzyme. So attaching this and then having a conjugated enzyme makes it very sensitive. This is what they call enzyme-linked immunosorbent assay, or ELISA. So most antibody assays are ELISA assays. This is what we use. But in the case of this coronavirus, you can detect it only when the body produces antibodies against the coronavirus. So there is a lag phase. As soon as you're infected by the coronavirus, it does not immediately produce enough antibodies. It has to sort of get itself settled and reproduce in large numbers before the antibodies become detectable. The real way of detecting the presence of the virus is what is called an RT-PCR assay of nasal swab extract of the virus RNA. This is what they call a PCR assay, polymerase chain reaction. This is an interesting concept that Kary Mullis, and he won a Nobel Prize in 1993 for this. He found out if you take sort of a DNA and if you heat it, it separates. If you heat it between the 94 to 98 degrees, it separates. Then if you cool it to 50 to 68 degrees, you can have pieces of DNA that you put in that will go and anneal itself, stick to itself. And then if you put the temperature at 70 degrees, it fills in the empty spaces. In other words, there's synthesis of new DNA. This is what happens when you have a mixture of nucleotides, DNA primers, and you put it in this PCR cycle. And this will go on and on, making thousands of copies. So you take a small piece of viral RNA, convert it into DNA, because you need the DNA to do this. And that DNA, when you produce this DNA, you convert RNA into DNA using reverse transcriptase. I told you about the reverse transcriptase that the HIV virus, all these things have. That has been sort of, you can buy this now from companies and you can use this. So you make a large number. That way, you can have this RNA amplified. And then when you amplify it, you can detect it. And this is a PCR machine which you use, polymerase chain reaction. But of course, this is an involved process. It takes time. And you have to be careful because RNA is very delicate and breaks down very quickly. So you have to have protective things put in there so that RNA doesn't break. And so, although that's why there's always a delay, because they say not all labs can handle it, because this is a sophisticated machine. And I remember I used to use it when I was working in Seattle in Lynn Rediford's lab at Washington University. There, sometimes it will work, sometimes it won't. So It's a sort of a temperamental machine. Now, with people who are well-trained, they can do a good job. So you have well-trained people using this. But things have changed now. I'll tell you a little later about that. Now, this April 19th, an amazing new invention that's going to increase testing dramatically was approved by Health Canada. And it's made in Canada, in Ottawa. It's called the RT-PCR Cube. It's the size of a coffee cup. Can analyze nasal swabs and provide COVID-19 test results in 1 hour. And it's 100% accurate and can be run by anyone. Paul Lem, the CEO of Spartan Bioscience of Ottawa, is an alumnus of the University of Toronto. Health Canada has approved the test, and the units are going to be mass-produced and shipped all over Canada. The RNA in the swab is extracted, reverse transcribed into DNA, annealed with primers for a region of the virus DNA, and amplified. Fluorescent dye bound to the probe is released at the end of the reaction, and fluorescence is measured fluorometrically. The protocol for this machine has been published by CDC, the Centers for Disease Control, and is publicly available. So he used this machine and tailor-made it to suit COVID virus and miniaturized it. And I think this is going to revolutionize our testing scheme. Now, we have heard all kinds of drugs that are available for control. Now, there are numerous claims of drugs that can be used for treating COVID-19 infection, but none of them have undergone rigorous clinical trials to establish their activity and safety. Currently, many are undergoing tests, so we probably will have treatments available soon. So right now, they are all at the anecdotal stage. We don't have anything established. A few of them have a lot of promise. One is an antiviral drug called remdesivir. An antiviral drug has had a lot of promise, and many severely sick individuals in respirators have recovered. But the problem with that, it has to be delivered by infusion. So you got to have a drip, and you have to be infused this over a few days. Similarly, there is another antiviral drug called favipiravir, an antiviral drug being developed in China, and it is in advanced state of testing and is also showing great promise, curing people who are severely affected. We had a lot of hype about hydrochloroquine, an anti-malarial drug also used for treating lupus, has been touted as a great drug, but has not lived up to its expectation. The only country that has made money out of this is India. It has shipped massive amounts to Russia, to US, and even to Canada. I don't know why we bought this, because it doesn't work very well. But of course, there's a sucker born every day. The next is a very interesting concept coming out of Israel. It's called pluristem therapeutics. It uses human placenta and harvests some smart cells and programs these cells to secrete therapeutic proteins in the body of sick people. It has just treated its first American patient after treating 7 Israelis. All of them have recovered. The success rate is very high. Again, we haven't got rigorous clinical trials. Now this one, they can inject these cells into the muscle, and it goes and establishes itself and secretes the protein which targets the COVID virus. Then you have all types of other things. There is AT100 by Airways, TZLS monoclonal, OYA1 by OYAGen, PPI002 by Beyond Spring, Ifendopril by Algermon, BX21 by Oxfam, MERS vaccine, Novavax, Actemera, Roche, Galsdevir, Biosis, Region 3048 and 3051 by Kevzara, SNG001 by Synergen, Amnioboost by Lattice Biologics, A1-based therapeutic by Mation, and so on and so on. So there are lots of things in the works. With the advanced technology available using artificial intelligence and in silico designing, where you design everything on the computer, we will soon have drugs for COVID-19 treatment. But must understand, the real answer is development of a vaccine leading to herd immunity is, however, the ultimate solution. We really do not know how vaccines are going to pan out. We are in uncharted territory. The most optimistic view is that it will be like measles vaccine, providing us with one lifetime protection. On the other hand, which is like the flu vaccine, it will provide a lukewarm protection for a season. With the panoply of vaccines under development, we can hope for the best. Coronavirus 19, although an RNA virus, has shown limited amount of mutation, which is a good sign. Several components of the coronavirus 19 have been used as antigens to make antibodies. Also, animal and plant platforms are being used to mass produce the vaccines. List of vaccines under development: DNA vaccine by Fusogenics, Shadox vaccine at Oxford University, AdCovid Ultimex, TJM2 at OneMab Pharma, VLP by Medicago, intranasal by Altimune, INO 4800 and INO 470 by Inovio, Hong Kong vaccine by University of Hong Kong, vaccine generics by Vaxi, Serum Institute of India, DNA vaccine by Zydus, monoclonal antibodies by biotech. These are but a few that are in the works. So sooner or later, we will come up with a vaccine. With the advanced state of vaccine technology and the numerous trials around the world, we should be able to get a vaccine that works. Vaccine development takes time, and it'll at least take a year before all of the safety tests and clinical trials are completed. But it is the ultimate solution to herd immunity. Now, I have made a pandemic prognostication, or wishful thinking, I call it. What's going to happen with all these things that are happening? Governments are pouring millions of dollars. Social interests have overtaken everything else. So we can make some conclusions. One is importance of a responsible and empathetic government will become paramount. We need a government that responds to the people's needs, and we need empathy, and we need support, and something that's not happening south of the border. Second, the plight of the have-nots has been sharply brought into focus, and social changes to redress the situation will probably occur. Look what happened in all these, people at the retirement centers, and their old people are. And, they have had very shoddy places with very poor sort of protection. And look at all the people, homeless people. Look at the prisoners in the prisons. They are all sort of foci for the infection. So we will take a hard look at it and see that they are afforded protection. Third is self-reliance for essential care products will be addressed. We are going all over the world buying stuff. We don't have to. We should be able to make all this here. They are not really, sophisticated things. They are very simple things. But unless they are made and available, we are going to expose all the, frontline workers to the COVID infection. Next is a quick response mechanism to any future pandemic will be in place. I heard that during the Obama presidency, he had a rapid response team in place, which was dismantled by the next president. Now, we need to have something like this in place so that should something like this happen, and it will happen, because this sort of zoonotic viruses all over, and we don't have to play this blame game. It can come from any place. It can come up from our own backyard. So we have to be careful, but we need a mechanism to address this. Then there will be a trend towards democratic socialism, similar to the Scandinavian countries. Equitable taxes to eliminate homelessness, housing for everyone, subsidized, well-run, affordable retirement homes, adequately funded education systems, healthcare is expanded and well-funded, guaranteed income, and universal pensions to all are a few to be considered. And then we will face the new normal. The new normal will definitely be very different. It's never going to be like the good old times. The challenge for us will be to happily adapt. Thank you.
Hey. Thank you. Muted, everybody. Wonderful. We can applaud.
Certainly will.
And Arthur, did you want to take some questions?
Yes. Now I love to get this off the screen, and then I will be able to see people.
So you should be able to see hands or virtual hands.
There they are.
There we are. There we are. Okay.
Well done.
Oh, thank you. Thank you.
Well done, indeed.
That was very well done.
Thank you.
Oh, what happened there?
Good. Basil.
Yeah. Arthur, when we talk about zoonotic transmission,
Yes.
these viruses come in from animals.
Yes.
I understand that there are viruses that have been discovered that are much more deadly than COVID-19. So, have you been studying any of these in your academic life?
Now, what the thing is, we always say that viruses, the highly adapted viruses, are those that don't kill their hosts.
Right.
They just give them a mild type of problem so they can coexist. If a virus goes and destroys the host, it is going to kill the source of its own survival. So like Ebola, for instance, deadly virus, but it was killing everybody off left and right. So it's not going to survive. So progressively, when viruses evolve, they reach a stage they can happily coexist. We know that because we were working on the spruce budworm virus in the lab where I was working. And the spruce budworm virus just kills a certain amount of spruce budworm, not all the spruce budworm, and so that it can survive. And we were working on the virus as a method of control, and we were doing some recombinant changes. We were inserting an insect gene into the spruce budworm virus and infecting it to the insect.
And Dave Hardy, he's got a question.
Dave?
Hi, Arthur. First of all, thank you very much. As usual, your presentations are incredible. I always learn a lot.
Thank you.
I want to go to the herd immunity, and I'm thinking of the polio virus.
Yes.
Where once we had a cure, it took 50 years to get to that herd immunity, or we're not there yet. Is this something that we're looking at in the future for COVID-19?
Yeah. No, the thing is, herd immunity is going to be acquired progressively over the years. Because if we have a vaccination thing, okay, let's say there's a good solid vaccine that has been developed. And in Canada, we go ahead and vaccinate people across the country from one end to the other. Now, this would give us the herd immunity that we need. But this may not be enough. We may have to go across globally and do it all around the world, because this is no time to confine ourselves to our own country. This is a global problem, and and we have to address what's happening in other countries. Some of the poor countries may not be able to afford this. We may have to subsidize and provide them with a vaccine. So this is an important thing. That's where we can get this herd immunity. But it's not going to be done overnight. It's going to take a while. You're right, because it took quite a while for this polio to get this thing. But take chickenpox. We got herd immunity. It's sent all over the world. Smallpox, and it's distributed all over. And we have the vaccines for children. You get this measles, mumps, things like that. They are well protected. Although for chickenpox, we have this problem with shingles, that you have a shingles vaccine now. Okay.
Arthur?
Yeah.
Can I question? Hello?
Who is it?
Hans?
Hans. Okay.
Yeah, I'm just wondering, I look at the global statistics quite too much, perhaps, and I'm curious why it seems like Europe and now United States are the champions in the world, which supposedly are the richest countries, but it attacks the poorest parts of their countries, and yet places where I would expect to explode, like let's say India, or like they apparently seem to be doing very well by comparison. Is this just
That's a very interesting question. I was also intrigued by that. So I talked to a friend of mine. Who recently came back from Bangalore, and I said, "How is it in India where people are sitting on
up the wall?
Oh, really?
one another, and there's a whole lot of people all over the
If it
world."
comes up again. Hang on.
Okay, everyone should really unmute when somebody is talking. You hear everything, every cough and everything.
Okay, what happens is in India, they say the incidence of COVID-19 is very low. The number of deaths is very low.
Yeah.
And why? Is it because of the heat and the humidity? Not really. There are other places where they have heat and humidity. And they say India has had so many kind of diseases, so they have a library of antibodies built in everybody. And maybe
Could
some
be.
of these things protect them against COVID virus. One in particular is BCG vaccine. I remember when I was in high school, they had a sort of a blitz on vaccinating everybody with BCG for protection against tuberculosis. And almost everybody was tested. We had a skin test, and they would test whether you have antibodies against tuberculosis. And if you had, you don't need BCG. But if you didn't, they'll give you the BCG vaccine. And they suspect that this BCG vaccine may be protecting them against the COVID-19 virus. And this is totally anecdotal, hypothetical, but it's an interesting observation.
Okay, let's.
Good.
Gerd, I'm going to have to find Gerd here to unmute him.
That's okay.
There you
Just a very quick one. If you're on your computer and Barry has muted you, all you have to do is push down the space button, the space on your keyboard, and that temporarily mutes you while you speak. So it's very easy. As soon as you let go, you're muted again.
Okay.
So that's the one little trick. But the other thing is this, Arthur. Just yesterday, I read an interview with one of the foremost medical people in the UK. His name is Dr. Nabarro or something.
Yes. I read it too. Yeah.
And basically what he says, if I understand it correctly, is that he is very doubtful that there will be ever a vaccine that will work 100%. If I understand it correctly, he says it will be more like the flu, where we have a vaccine that's somewhat useful, but it's never totally useful. I don't know if that's a good comparison. Maybe you can comment on that, Arthur. I don't know.
Yeah, but the thing is, the flu virus mutates a lot. So you have, every time they have an antibody against the flu virus, the next season, the flu has changed. So it doesn't respond to this antibody. So what they do is they collect all the previous season, the flu components from around the world, and make a cocktail and produce antibodies. And they say, hopefully this will cover. And it covers and doesn't cover. It doesn't work very well. On the other hand, in the case of this virus, it has got a specific spike protein. And it needs the spike protein to enter into the cell. So if you target the spike protein, you will probably disable the virus. And if you can successfully do that, and if the vaccine, if the antibody stays in your body, that's the next question. You know, it may not stay that long. It may stay for a season, and then you may have to get it again. If it stays, then it'll be a wonderful vaccine. The proof is in the pudding. We will never know until these vaccines are tested. I saw the Navarro's account. That's a one end of the spectrum where you're very pessimistic. On the other end, you can be optimistic, and you can have this herd immunity. It's Gordon.
Yeah, on CBC radio this morning, there was, I'm not sure exactly what it was, but obviously some kind of scientist was being interviewed. And he said something that surprised me. He said that somebody can transmit, if that's the right word, the virus to you just by, as he put it, exhaling or just breathing on you rather than you know, actually coughing or sneezing, which kind of surprised me. What do you think on that?
Well, several people have made this comment that they say, you know, if they're close enough, and you know, when they sort of speak to you, invariably, as Trudeau put it, a moist speech, you might be able to transmit the virus. It is not it's not aerosol transmission. That's what they say. You know, it's not like SARS. Like one person coughs in the subway, and everybody covers their face from one end because it is transmitted in the air. There is no evidence for that. It has to be close enough so that there will be some direct particles moving to the other person. So I'm not so sure about that. The consensus is you have to have this, you know, little droplets that you get, you know, you breathe in. not the aerosol. Dave.
Thank you.
Unmute. Arthur, just to question your comment about the changes to our social systems and politics and so on in the future. So I'm looking at Sweden, which is one of the most democratically socialist countries out there, has 10 times more cases and deaths than its Nordic neighbors, and is one of the highest number of deaths per capita per 100,000 people in Europe. So why, we talked about why India is kind of not getting as much. Why is Sweden getting more?
Well, Sweden tried a different kind of experiment. Sweden and Britain initially. What they did was, okay, we're not going to do anything about it. Let it come and sweep the country, and then people will develop immunity, and then they'll be okay. What they forgot to put in the equation was, it is going to kill all the vulnerable people, the old people, the people with the pre-existing conditions. They are all going to be weeded out. Now, that is not socially acceptable in our kind. There is a certain amount of callousness involved. You're doing a sort of a Darwinian type of thing. Let it come, survival of the fittest, and let the survivors survive. All the others, sorry, it's goodbye Charlie for you. Now, that is an experiment that I think Britain tried. Now they're quickly trying to withdraw themselves. Whereas in India, they did have a lockdown, a severe lockdown. I saw people being, police coming and taking little sticks and whacking people on their back and chasing them in, pushing these, in this putt-putts. They put a stick to the driver and push him out of the putt-putt, and all these crazy things happening. I was talking to my niece in Bangalore, and I asked her, I wanted her to go to my wife's grave and place some flowers because there's the anniversary. He said, Uncle, I cannot leave the house. There are police patrolling every place. So she couldn't leave the house. So they are delivering food to people. So they did try. I mean, it's a difficult thing to do in India with this population and the concentration. And also, strange things is, people who have, household help to do the cooking and everything, apparently they're all not allowed now. And these people are trying to return to the villages. And when they come to the villagers don't want them. They say, don't bring your COVID virus to us. So these people are, in between, from the city and the village. They hang in there. So it's sort of a strange problem.
Are there any more questions?
Danny?
Yeah, just to be the devil's advocate here, what you were talking about Sweden and Britain and the differences between them and India, the possibility of massive famines because of what we're doing right now. What's your take on that?
I really don't know how it's going to affect us, but most of these farmers isolate themselves in their normal everyday living anyway. You see, in Canada, every 5 miles there'll be one farmer, living there. So he's going to isolate himself anyways. So I do not know.
Yeah, well, I'm really talking about the effect on the poorer countries in the world.
Yes.
Who have their problems anyway. We hear about farmers here plowing over fields because the price has gone down too far, pouring out milk down the drain because the price has gone down too far, and that's what we're doing in these countries, the rich countries. How do you think that's going to affect the countries who could be entering into famine?
You may have a sort of a transient famine, but I think they'll soon come back. You may have in the immediate future, you may get spots where there's famine, but I think in the long run it'll sort of work itself out. That's
okay, thank you. Thank you.
Dave.
Dave, you have to unmute. There we go.
Just to bring it all home, next week we have John Stapleton, who's a kind of a Canada reputation-wide researcher talking about the effects of the coronavirus on North Scarborough. And it should be very interesting. I've seen his blog and data, so I just thought I'd give you a heads up. Next week's meeting should be quite interesting. Thank you.
Yeah, thanks, Dave.
I would like to add one more thing. My son, as you know, works at Mount Sinai, and they have all been issued, letters of warning saying that they can be called upon any time to go into the wards and look after these COVID patients. But he says lately there have been a scale down in Mount Sinai because there haven't been too many people, admitted, and they sent a letter saying, you guys are okay, we don't need you anymore. So it's a good sign. And also, Lunenfeld Institute, it's a part of Mount Sinai. They are developing a antibody assay, and apparently it's a very quick assay, and they want to test it on using blood, that you get this pinprick blood. So they heard about diabetes people collecting blood with pinprick. So they asked my son to collaborate with them, with his team of pinprickers, with the Lunenfeld people who are developing the antibody test.
Okay, so I'm going to ask Al Meredith to thank our speaker. Al?
Can you hear me now?
Yep.
Okay, well, Arthur, the first thing I'm going to say is that I went to a different school to you because my science teachers and chemistry teachers, I don't think I got past 101 because you speak at a different level to the rest of us, I'm sure. But understanding that you're a very knowledgeable person and an excellent, excellent presentation today. What can we say about you? You're such an excellent presenter every time you speak to the club. And I always seem to get the job of thanking you, which I'm not really qualified to do, but thanks again, Arthur.
Okay, and I'm trying to unmute everybody so we can follow up on that.
Thanks, Arthur. It was a great presentation.
Yeah. Thanks, Arthur.
Thank you. Thank you very much. Thank you.
Arthur.
It's amazing the speakers we have in our own club. Some of the best speakers in the world there.